question. You needed to identify segments A, B, C, and D from this pressure volume loop, right? And I have another question for you. Can you all see my pointer as I'm working?
Yep.
You should be able to.
No, actually, yes.
Last class, I could see it, but it was like so lagged. It wasn't helpful.
It wasn't really in real time.
It was like almost a whole slide behind last class at least.
Oh wow. Okay. That's not something I think we can fix right here. So I'll try figuring that out as well. So in the pressure volume loop, these would be the answers I would expect on a quiz or an exam or something of that nature. All right. And I'm not going to belabor this pressure volume loop this morning because we have to keep moving in renal because I want to get at least 80% of the way through renal before the end of the week. So we ended up last Friday talking about the fact that the kidney is really responsible for pressure volume, arterial pressure and body fluid volumes, extracellular fluid volume regulation, and intracellular fluid volume regulation. We talked about how you can calculate intracellular and extracellular spaces. Now these aren't precise calculations, these are estimations. Total body mass is 2/3 of total body water is 2/3 of total body mass. 2/3 of total body water then is intracellular fluid. All right, and so your kidneys are responsible for maintaining your cell volumes at all times. Then what's not intracellular fluid volume has to be extracellular, and the extracellular compartment is made up of fluid that's interstitial, in other words, between cells, and fluid that is in the vasculature, otherwise known as plasma. And of your extracellular fluid volume, 80% of that extracellular fluid is in the interstitial space between cells, and 20% is in the plasma, makes up the plasma or the vascular volume. And I showed you these figures that sort of pictorially depict total body water, which is the outside rectangle, intracellular volume shown in blue, and then the extracellular fluid shown in green, and extracellular fluid, 80% of that is interstitial, shown in the green, and then in the yellow is plasma or vascular volume. Now we're going to control the fluid movements between each of these body fluid compartments by osmotic forces, primarily sodium chloride, but that would also include potassium. That would also include bicarbonate, calcium, glucose, small proteins, anything that is small enough to be soluble or dissolved in the plasma makes up the forces that are going to move fluids between the body fluid compartments. Now we also have hydrostatic forces, and that's just pressure. So the hydrostatic force of the vasculature we've been talking about. That's simply blood pressure, right? That's blood pressure. So that's a hydrostatic pressure that's going to move fluid from the capillaries out into the interstitium, out into the intracellular space. We also have oncotic forces, or proteins. And proteins are sequestered and maintained within the plasma. They're kept and held inside the plasma. So that's going to be a force that moves fluid from the interstitium back into the vasculature. That oncotic pressure, that oncotic force of the proteins in the vasculature. Now, we ended up on Friday talking a little bit about how different organisms regulate their body fluid volumes, and we have two different kinds of body fluid volume regulators in the animal kingdom. One is an osmoregulator. That's what we are. That's what mammals are. That's what most animals are. Most mammals are. They're osmoregulators. In other words, as our environment changes, changes around us, whether we're in the desert, whether we're in the rainforest, whether it's hot, whether it's cold, depending on what our environment is, whether we're swimming in the ocean, whether we're swimming in fresh water, depending on what our environment is, we regulate our internal milieu. We regulate what's inside of our body fluid compartments ourselves, and we regulate that almost entirely by regulating kidney function. How much fluid do we allow to excrete through our kidneys? How much fluid do we retain? How much sodium chloride do we retain? How much sodium chloride do we excrete? We do all that by regulating our body fluid volumes and our body fluid components. Now, there are other organisms that are osmoconformers. So in other words, they conform their internal osmolarity. They conform their internal body fluid compartments to their environment. And I used the example previously of an organism that lives in an estuary. So as the tide comes in an estuary, that animal, that organism is exposed to very high salinity environment, high sodium chloride environment. It's about 4 to 5% in that environment. They will change, they will conform their internal body fluids to match that environment. Then as that tide moves out and fresh water may move in, now all of a sudden their external environment has changed a great deal. Those osmoconforming organisms will get rid of salts, they'll get rid of sodium chloride and conform their cells. They have evolved in a way that they can conform to that external environment. So we have all different ranges of different kinds of organisms that exist within their habitat as being osmoconformers or osmoregulators. Now this slide just shows you some of the differences in internal sodium chloride concentrations of a number of different organisms. This is the composition of the extracellular fluid that these organisms live in. So seawater is about 460 millimolar of sodium. So if you're a jellyfish living in seawater, all right, they live in about 460 millimolar sodium chloride. Their internal milieu is about 454 millimoles of sodium chloride. If you or I had an internal concentration of sodium chloride at 454, we would be in big trouble. All right? And as you can see, as you move down different organisms in seawater and freshwater, let's look down here at mollusks. If you're an aplysia, all right, or a squid, a sea slug or a squid that lives in salt water, your internal sodium concentration is very, very high. They must be osmoconformers. In contrast, freshwater clams, their internal conformation, they conform to an internal sodium chloride of only about 16 millimolar because they don't have any very high sodium concentration in their external environment. So this shows you how these organisms are able to vary and change their internal sodium chloride concentration depending upon the environment in which they exist, in which they live. Now, what do you notice about potassium concentration over here? So they can conform their internal environment to the sodium concentration, but their potassium remains relatively constant regardless of what the external environment looks like. And that's because we've learned in terms of cell biology and cell physiology that we have to maintain intracellular potassium concentration fairly constant. Now, some organisms, like the squid, they've evolved so that they can deal with 20.6 millimolar potassium in their internal environment. If our potassium concentration inside of us gets up to 10, 11, or 12, we go into cardiac arrest. We're in big trouble. But other organisms have evolved in different ways. So we're also going to talk about, in terms of kidney function, how we not only control sodium in the internal environment and concentration of sodium chloride, but also that of potassium and potassium chloride. Now, remember I told you about us in terms of electrolyte composition of human body fluids. I wanted you to know and understand and know the numbers for sodium and potassium concentration of serum or plasma. And we know that sodiums were around 140, potassium were somewhere between 2 and 5 or 6 milliequivalents per liter of water. We now have to add the anions onto that group. And I want you to know and memorize the concentration of chloride, which is about 104, and the concentration of bicarbonate, which is about 27 milliequivalents per liter. What do you notice if you add 104 and 127? What do you get adding 104 and 127? You get about 131, right? About 131 millimolar for the anions in your serum. What do you notice about sodium? It's 142. So we are mostly composed of sodium chloride and sodium bicarbonate in your serum. You're mostly sodium chloride and sodium bicarbonate. Now we do have a gap. We have about a 10, 11, 12, 13 milliequivalent per liter gap in anions between chloride and bicarbonate and sodium, don't we? And we're going to talk about that anion gap when we get to acid-base balance. When we get to acid-base balance after exam 2, we're going to talk about that anion gap. But for now, you must know the concentrations of sodium, potassium, chloride, and bicarbonate in the plasma and interstitial, extracellular fluid. Okay? And we're going to talk more and more about how we regulate chloride, how we regulate sodium, and how our kidneys regulate potassium in our extracellular fluid. Okay? All right. Homework problem. I want you to do this problem. You're a world-class exotic animal veterinary anesthetist, and Sydney calls you up to anesthetize their rare Bengal tiger collection. You're concerned because this tiger is elderly and somewhat thin, and you need to calculate that animal's extracellular fluid and blood volumes. If that tiger weighs 345 kilograms, what is the estimated extracellular fluid and blood volume? Now, I've given you the extracellular fluid volume and the blood volume, and your homework for this week, for between now and Wednesday, is to calculate those differences in extracellular fluid volume, to calculate how I came up with those estimated extracellular fluid volumes and those estimated blood volumes, and we will go through that calculation first thing on Wednesday, okay? But you need to do this homework, please. You need to think through it. Does everybody hear me? I need you to think through it so that you understand how I got to these values. And I don't know. I sometimes throw you a curveball, don't I? I sometimes put up wrong answers. So I want you to go back to this slide and calculate and figure out how we got to these values for that Bengal tiger. All right? Because I'm quite certain the Sydney Zoological Society would want you to not mess up your calculations in terms of understanding what that Bengal tiger actually had in its extracellular fluid. All right, so now here are some organisms that tell you a little bit about how these organisms regulate their internal milieu. All right, so it gives you the type of animal, shows you a bunch of different different number of animals. So let's take the marine mammal, for example, whales, the porpoises, and so on, those marine mammals. How do they regulate their internal milieu when they don't drink seawater? Right? They do not want to drink seawater. But somehow they have to maintain a urine concentration relative to blood that's either isoosmotic, hyperosmotic, or hypoosmotic, all right, or hypoosmotic. And this tells you their blood concentration relative to their environment and their urine concentration relative to the blood. And this will tell you a little bit over here on the right how they go about regulating their intracellular and their extracellular fluid volumes, whether they're a marine teleost, all right, a marine fish, a freshwater fish, whether you're an amphibian that goes between the land and the water environments where they live, et cetera, et cetera. What if you're a marine bird, all right? What if you're a marine bird and you drink seawater? How do you get rid of that excess salt by drinking the seawater? What do you do? Well, they have salt glands in their nose to get rid of their salt. So we've evolved throughout the animal kingdom in many different ways to regulate our sodium concentrations and our extracellular fluid in order to keep our body fluid volumes constant. Now, what about us? What about humans? You're floating at sea, and seawater is somewhere between 3% and 5% sodium chloride, salt. Your urine can put out about 2% salt. Can put out about 2% salt. So if you're floating out here in the sea, is it a good idea to drink this seawater? Would that be a good idea?
No.
It's not a good idea because what's going to happen is you're taking in more salt than your kidneys can put out. When you're maximally excreting sodium chloride from your kidneys, you're still retaining about 1% of that salt that you would be drinking out here in the seawater. So you would be better off to hydrate yourself by drinking your urine than you would your salt, your seawater. Because if you drink your urine, you just recycle and excrete that salt, and you now may retain some of that water that's in that urine. If you drink the seawater, you're just going to dehydrate your cells. And you need to understand why that's the case by the movement of body fluid volumes. Okay? So the fundamental problem that these people would exist if they're stranded at sea, their fundamental problem is, where do I get water? How do I get water so that I can maintain my internal milieu? All right. We can survive at sea without food for quite some time, 30, 40 days. We can't survive even a week with no water. What's the Tom Hanks movie? What's the Tom Hanks movie where he's stranded on the island, his FedEx plane crashes?
Castaway.
Yeah, Castaway. And what does he do when he's floating on his raft? He makes all kinds of different ways that he can collect rainwater so he has water to drink. He concocts a whole bunch of different ways to collect the rainwater and save it and store it so that he and Wilson have plenty of water to drink in order to survive. Because he knows he can't drink the seawater, right? Can't drink the seawater. Okay. Now, different organisms have evolved in different ways to deal with this issue. I'm just going to show you one example of seagulls. They're actually gulls, but they're gulls that live by the sea. And they actually drink seawater, all right? They actually drink seawater. And their kidneys have evolved so that they can put out about 3% salt. But that still keeps excess salt inside their bodies. And so now they have salt glands in their nasal region that actually concentrate that sodium chloride in these centralized canals, and they spit out little crystals of sodium chloride onto their beak so that they can maintain their internal milieu constant and get rid of the salt that they drink. So different organisms have learned how to. Now, would you call this? I want to ask you the question. Would you call this animal an osmoconformer or an osmoregulator? Are they conforming to their environment, or are they regulating their internal salt in their environment?
They're osmoregulators.
That's right. They're regulating their salt. They've developed a different kind of technique. We can't spit crystals out of our nose, right? We have to count on our kidneys to get rid of our salt. So those seagulls have become osmoregulators by developing salt glands within their nasal region to collect the salt from their plasma, from their extracellular fluid, and put out sodium chloride crystals. Now, all different kinds of organisms have different ways in which they control their internal sodium chloride environment. Here's your local laboratory crayfish, all right, and they reabsorb water in a labyrinth. They actually have their kidney and genitourinary tract functions within right behind their eyes, between the thorax and their head. And they have urine that they put out the top of their head. They have a bladder, and they have an organism or an organ within their body that's a labyrinth that reabsorbs water back into their body. It's sort of an end sac, and then they concentrate their urine and they put their urine out through their nose. You have different kinds of organisms that have nephrotidiophores. There are conducting tubules that have nephridiophores that they're able to control their internal milieu of their environment by excreting salt and water through their skin and through pores that connect in their skin. So different organisms have evolved and developed different ways to control the amount of salt and water that they have and they excrete. And we have animals that live in the desert, such as the kangaroo rat or the desert rat. And these animals during the daytime have adapted behavioral adaptations to live underground where it's cool. They eat seeds that have free water in them, and they can concentrate their urine to the point where they literally put out no water in their urine. They have dehydrated feces where they just put out little pellets that have virtually no water in them. And in fact, these kangaroo rats can excrete urine with concentrations of about 9,000 milliosmoles per liter. They actually don't have to drink water at all. They get enough water through the eating of seeds and the metabolic production through oxidative phosphorylation, where they generate CO2 and water through oxidative phosphorylation. And that water is enough to maintain and sustain them because they can concentrate their urine to very, very high osmolalities, an osmolality of 7, 8, 9,000 milliosmoles per kilogram. We can't do that. As humans and mammals, most of us can only concentrate our urine to about 1,500 or 2,000 milliosmoles per kilogram. All right. So now, where do we, in the kangaroo rat, where do they get their water and where do they excrete their water? Well, where do they take water in? Where are their gains? 90% of their water comes from their metabolic production of carbon dioxide and water through oxidative phosphorylation. They get about 10% of their water in their food, none of it through drinking. So their intake is 100% from the food that they eat and the water that they produce through oxidative phosphorylation. Now they lose water through about 70% of their water is lost through evaporation and perspiration. And we're going to talk about how we lose a lot of water from our lungs and evaporation every single day. We as humans, we lose, thank you for hydrating yourself, Lindsay. We keep ourselves very well hydrated, all right, because we lose about 20% of the water we take in as mammals just because we respire every single day. We lose about 25%, so the kangaroo rat only loses about 25% of its water through its urine. So most of the water that we lose is through kidneys, and about 20% of it is through our perspiration and our evaporation. Whereas the kangaroo rat has exactly the opposite situation. They lose 70% of their evaporation their water through evaporation and perspiration, and only about 25% through their urine, because their kidneys are able to reabsorb so much water. And we both lose about 5% of our daily water intake through our feces, making 100% of our water loss equal to our gains. Water intake and water loss have to be the same every day. Water intake and water loss must be the same every day. Otherwise, we're blowing up like a Michelin person, or we're shrinking down to nothing. All right? If you go out and exercise very hard, you go for a 5-mile run on a very hot day. If you're hiking out of the Grand Canyon on a very hot day, you must take in a lot of water, because you're going to lose a lot of water through evaporation and perspiration. So the amount of water we take in and the amount of water we lose every day have to be equal. They have to be the same. So our kidneys spend a great deal of time they spend most of their time regulating and maintaining body fluid volumes through extracellular fluid and intracellular fluid. And we do that by moving fluids between these compartments, between the vasculature and the extracellular fluid, by different forces. Osmotic forces that are primarily handled with sodium chloride, oncotic forces, which are proteins, and hydrostatic forces, which are blood pressure, interstitial fluid pressure, and intracellular hydrostatic pressure. Remember, when we talk about hydrostatic pressure, that's just like pressure in the air in your tires. That's like pressure in your blood. That's like pressure in your interstitium. If we move a lot of fluid from our vasculature into the interstitium, hydrostatic pressure between our cells goes up, and we're going to push fluid into cells as well as push fluid back into our vasculature, as long as that pressure in the extracellular fluid exceeds that in the venular side of the capillary. So now we're connecting the vasculature to regulation of body fluid volumes and ultimately the long-term control of blood pressure. Now, I have another homework problem for you that's really a hard one, but I want you to work on this and try to figure it out between now and Wednesday. So if you want to inject a relaxant such as Demerol to get a blood concentration of about 20 milligrams per milliliter, how many milligrams of Demerol would you administer? Now you have to assume that the drug distributes evenly throughout the blood and the vasculature. And you're going to do this for a vascular volume that's about 4.37 total liters. So this would be in a human with a vascular volume of about 4.37 liters, and you want your Demerol to be 20 milligrams per milliliter. So that means if you're going to get 20 milligrams per milliliter, you would have to inject 87.4 grams of Demerol into that individual to get that concentration of Demerol. I want you to work through this problem, show the math, understand how you reach these values. I've given you the answers. I need you to figure out how you're going to calculate that volume. Because when we get to next Monday in our exam, you may have to calculate one of these concentrations given the vascular volume, or given just the total body mass, and you have to calculate the vascular volume and then figure out how much of a given drug or compound you'd have to inject to reach the stated concentration in the plasma. Okay? This is kind of an important calculation for any and all of you. All right. Let's start out now doing some basic kidney anatomy. All right. The kidneys are two small shaped organs looking like kidney beans that exist in your the lower back region of your thorax. They're in your abdominal cavity, not your thorax, within the abdomen. And the kidneys that you see here, they lie outside the peritoneum. They exist retro outside of the peritoneum. So your whole abdominal cavity is encased in a tough connective tissue called the peritoneum, and you hold your intestines and your liver and your pancreas and your spleen and all of your organs in your intestinal cavity, your stomach, et cetera, sit inside this bag called the peritoneum. Your kidneys sit outside that peritoneum. They are outside the bag that the rest of your organs sit in, which is kind of handy because if you want to access the kidney to do a transplant or to do a surgery, you can simply enter through the back region in the back and go in. You never go through the peritoneal cavity, and you prevent infection by going in that way through the back, and you can access your kidneys in that way. The adrenal gland in humans sits right on top of this the one pole, the anterior pole of the kidney. In other organisms, the adrenal gland sits slightly outside that area and is not connected to the capsule of the kidney. So the kidney is encased in a connective tissue capsule. You have two major regions. One is the cortex, or the outside of the kidney, and the other is the medulla, or the inside of the kidney. And we're going to talk about the functions of the cortex and the medulla at length and how they are important for regulating body fluid volumes and regulating excretion of sodium, potassium, chloride, bicarbonate, etc., etc. All right? That's why you've got to know the concentrations of chloride and bicarbonate. We're not going to talk a great deal about calcium in terms of how the kidney handles calcium. That we save that for later courses, like BIO-350, where we talk at length about the hormonal and regulation of potassium or calcium excretion by kidneys and kidney function. Now, the filtrate of the kidneys ultimately end up being dripped into this pelvis of the kidney. It's a connective tissue area that ends up forming the ureters. And as you can see on the left, the ureters connect to the bladder from both the right side and the left side. Now, the kidneys are perfused by two arteries that branch off of the aorta, a single artery that branch off the aorta, and they're drained by a vein called the renal vein that drains directly into the inferior vena cava. The aorta connects to the renal artery, which then branches inside the kidney off to a number of pyramids, renal pyramids that you see here. And in the human and many mammalian kidneys, you have multiple pyramids within each kidney. If you're a rat or a mouse, your entire kidney is made up of a single pyramid. But humans and other mammals, horses, dogs, cats, hogs, humans, we all have multiple pyramids in our kidneys. Rats and other small rodents only have a single pyramid. It's these pyramids where we concentrate our urine and control our water excretion, the excretion of and maintenance of total body water. Now, the renal artery is, we have a single renal artery that branches off the aorta, as you see here. Now, in some people, you may have, during embryologic development, you may have two renal arteries coming off that aorta. And that's perfectly normal and happens in a significant percentage of people. And it just means that these initial branches never merge together during kidney embryogenesis, and you never reach this single renal artery stage. So you can see now the cortex that you see on the outside of the kidney and the medulla, which is the deep portions of these renal pyramids that extend down deep into the inner portions of the kidney, and they're responsible for the concentrating mechanism of the urine. Now you have some basic terminology that you have to be very, very familiar with and understand. So blood comes into the kidney, and it has a capillary bed called the glomerulus. The glomerulus, right here, you can see in this picture, it's a capillary bed called the glomerulus. You see it right over here. Here's that word. It's a large segment of capillaries that are surrounded by a group of epithelial cells that make up this capsule called Bowman's capsule. So this entire glomerulus that you see here has three major functions. It filters plasma, and we call that filtrate the glomerular filtration rate, and it has a fluid concentration that comes from the plasma. If the osmolality of your plasma is normal at 285 milliosmoles per liter, the concentration of the filtrate is 285 milliosmoles per liter. So your glomerular filtrate is just an ultrafiltrate of the plasma. We're going to talk about that more in a little bit. It'll probably be on Wednesday. You then have three other functions that I want you to be very familiar with. Secretion. Secretion. Anytime I use the word secretion, that means moving fluid from the capillaries into the tubule. If I say the word filtration, I'm moving fluid from the glomerular capillaries into the tubule. So there are two ways we get fluid into the tubule: secretion and filtration. Now we also have the next term, reabsorption. That's the movement of fluid and ions from the tubular fluid back into the plasma. From the tubular fluid back into the plasma. Then the last term is excretion. And that's urine flow rate. That's the excretion that comes out at the end of the day. That's the excretion that comes out at the end of the nephron and into the bladder. So we have filtration. We have secretion, that puts fluid and ions and particles into the tubule. We have reabsorption, which takes ions and water and particles out of the urine. And we have excretion, that's where we move particles, ions, and water into the bladder, into the urine, for excretion out the urethra. I failed to mention the last component of the genitourinary tract. We go from kidneys to pelvis, to ureter, to bladder, to urethra. And urethra goes from the urethral bladder sphincter, which then controls excretion of urine and tubular flow out the urethra into the exterior of the body. Now, we concentrate our urine, osmoconcentrate our urine by reabsorption of water late in the nephron right before the tubular fluid moves into the ureter and becomes urine. So we have renal plasma flow, that is the rate or flow of plasma into the glomerulus. We then have glomerular filtration rate, that's the rate of filtration from the plasma into the tubular fluid, and we have urine flow rate, which is the rate of excretion of urine out of the nephron into the bladder. Now, the kidney function, and we'll finish up with the kidney structures by talking a little bit about the cortex, the glomerulus, and the different structures of the nephron. The entire functional unit of the kidney is the nephron or the tubule. You'll hear me call it nephrons, you'll hear me call it tubules. Blood comes in through the renal artery from the aorta into the glomerulus, which is the first capillary bed. The kidney is unique in that its glomerular capillary bed is flanked on either side by an arteriole an afferent arteriole, and an efferent arteriole. It's nearly the only place, there's only one other place in the body you have this structure, and that's up in the hypothalamus, and we'll talk about that in endocrinology. But you have a glomerulus, which is a capillary that's flanked by an arteriole coming in and an arteriole going out. So therefore, we can control the hydrostatic pressure inside the glomerulus by constricting or dilating either the afferent arteriole or the efferent arteriole. Now, the tubule, the Bowman's capsule, which surrounds that glomerulus, moves into a proximal convoluted tubule, into a proximal straight tubule, then a descending loop of Henle, an ascending loop of Henle, then a medullary thick ascending limb, followed by a distal tubule, and that dumps into a collecting duct, which moves out to the urine. Now, many nephrons will dump into a single collecting duct. But each human kidney has about 1 million glomeruli and nephrons. The number of glomeruli and nephrons you're born with is what you have for life. We don't regenerate new glomeruli, we don't regenerate new nephrons. So at the end of embryogenesis, at the end of nephrogenesis, you have the number of glomeruli that you're going to have, you have the number of nephrons that exist for life. Now the last piece I want to show you before we end today is that notice how this nephron, after its loop of Henle, it folds back on itself to give you a juxtaglomerular apparatus. In other words, this nephron that's coming from its own glomerulus and its own Bowman's capsule can talk to the afferent and efferent arterioles and tell that glomerulus and that Bowman's capsule what's happened in that proximal tubule, in that loop of Henle, and in that medullary thick ascending limb in terms of sodium chloride reabsorption, in terms of water reabsorption, and in terms of how much tubular fluid flow is going to itself. So if you have too much tubular fluid flow going past itself, that juxtaglomerular apparatus will cause that afferent arteriole to constrict and decrease the tubular fluid flow. If you have, does this sound like a feedback control system, ladies and gentlemen? I hope so, because it is. If you have too little tubular fluid coming into that juxtaglomerular apparatus, you will then dilate your afferent arteriole and constrict your efferent arteriole to increase the tubular fluid flow and maintain that single nephron tubular fluid flow rate constant all the time. So you have sensors in this juxtaglomerular apparatus. You have effectors, which is the tubular fluid flow rate. You have integrators in this juxtaglomerular apparatus. And you have a controlled variable, which is changes in the afferent and efferent arterial resistance to regulate how much tubular fluid flow takes place in all 2 million nephrons every minute and every moment of every day. Okay, with that, we'll end for today. We'll pick up right here on Wednesday and begin with how we control tubular fluid flow and how the glomerulus functions and operates. And we'll talk a little bit about hydrostatic pressure and how we get filtration into the nephron, where filtration occurs in the kidney, and how we control glomerular filtration in each kidney. Please do your homework. I'm going to set the time for coming online earlier so we can get started closer to being on time for the next class. And I will figure out why we're not seeing the the posting of our videos so that you all have access to those videos that are being saved on the cloud. Any questions anybody wants to ask today? All right. Thank you all very much for attending today, and we will we'll take up there starting Wednesday and move forward on Wednesday. Take care, everybody. Stay safe. Stay isolated. Watch the silly videos of what we should be doing while we're that are posted on YouTube. That we should be keeping ourselves safe and healthy.
Thank you, Dr. Osborne.
Cheers, everybody. Thank you.
Thank you. Thank you. Keep us updated on your vaccine work. That sounds really interesting.